Hello students, today we will discuss on the topic anther and pollen culture and ovary culture. So what is anther culture? Anther culture is a technique by which the developing anthers at a precise and critical stage are excised aseptically from unopened flower bud and are cultured on a nutrient medium where the microspores or pollen grain within the cultured anther develop into callus tissue or embryos that give rise to haploid plantlets either through organogenesis or embryogenesis. That means in anther culture, the anthers after surface sterilization directly inoculated into the culture media of known composition, and during the incubation period the pollen grains or microspore present inside the anther bulb will multiply and ultimately form callus tissue or embryos, and from the callus tissue or embryos new haploid plantlets are developed. Next, what is pollen culture? The pollen or microspore culture is an in vitro technique by which the pollen grains, preferably at the uninucleate stage, are squeezed out aseptically from the intact anther and then cultured on nutrient medium where the microspore, without producing male gametes, develop into haploid embryos or callus tissue that give rise to haploid plantlet by embryogenesis or organogenesis. Next, let's discuss about androgenesis. So what is androgenesis? Androgenesis is the in vitro development of haploid plant originating from totipotent pollen grain through a series of cell division and differentiation. The androgenesis are of two types. Number one is direct androgenesis and number two is indirect androgenesis. So in direct androgenesis, the microspore or pollen grain behaves like a zygote and undergoes cells to form embryo, which ultimately give rise to a plantlet. However, in indirect androgenesis, the microspore or pollen grain, instead of undergoing embryogenesis, divide repeatedly to form a callus tissue, which differentiate into a haploid plantlet. The main significance of the anther culture or pollen culture is the production of haploid plantlets. As the pollen grains are haploid in nature, so when we culture the pollen grain or anther in a culture medium of known composition, then ultimately the plantlets will be haploid in nature.